Students, today we will start with the introduction to laboratory equipments. So we will start with the microscope first. In this video we will discuss regarding components and handling of a microscope, right? So aim of this experiment is to study component, life, microscope and its parts. Okay, so first we will discuss about base or stand.
So it is a U-shaped foundation which is used to give the stability to the whole instrument. Right, so as you can see here, this is the U-shaped foundation, and in this particular case there is a rectangular foundation.
Okay, so this is all about base or stand. Next, arm or handle.
So this is the arm or handle of the microscope, by means which it can be moved like this or it can be carried, right? And along with the arm, which adjustment instruments are attached, also the magnifying instruments are also attached.
Okay, then next is a stage.
Stage is the platform, or you can say the surface, which is used for the placement of the slide, which is having specimen over it, right? It can be moved vertically or it can be moved horizontally, right? It can be fixed kind of thing also. As you can see in this microscope, the stage is fixed. The magnifying objects can be moved, but stage is fixed. In this particular kind of microscope, the stage is moving. It is said to be the mechanical stage, right, which can be moved horizontally or vertically. These are the scales for that.
Okay, then next one, clips.
Clips, you can say it is a stage of removable clips or spring clips which holds the slide on its place.
Exactly. Then mirror.
It is a light reflecting device, as you can see here. It is used to concentrate the light in the condenser. But nowadays the concept of mirror has been depleting, but there is a concept of artificial illumination for which you can use the light, LED kind of thing, and this is the regulator for increasing and decreasing the intensity as per your requirement.
Right. Next one, that is a condenser.
As you can see, it is the system of lens which is placed under the stage, and it is used to focus the strong beam of light into the objective lens. As you can see over here, I will just remove and I will let you know what the condenser is. This is placed, this is the condenser, it is the lens system, right? What is its function? Its function is to focus the strong beam of light on the object which is being examined. Below the condenser, there is an iris diaphragm. It is the device which is present beneath the condenser and capable of manipulation of the light intensity, right? So you can adjust the light intensity by opening or closing of the iris diaphragm. So if you want to go for a stain preparation, you have to keep it on the fully open form, which is going to gather the maximum amount of light. But whenever you are going for the on-stain preparation, as for example the wet mount or other kind of preparations, you have to keep the condenser on the lowest light intensity. So you have to close the iris diaphragm.
Exactly. Then, okay, let's fix this. Okay. Then next one, body tube.
So as you can see, it is the hollow cylindrical tube. This is known as body tube, right? So it is a hollow cylindrical tube in which the light passes from objective lenses. These are objective lenses. Through the body tube, it is going to come out from the eyepiece, right? So at the bottom of the body tube, there is objective, and at the top of the body tube, there is eyepiece. So in this particular, you can see this is the hollow cylindrical tube, on the basis of which this is the objective lens, and at the top of the body tube there is eyepiece.
Okay, then nose piece and objectives.
This is the nose piece, or you can say this is the revolving disc. It is just to focus which kind of magnification you want, right? These are the objectives. So you can say this is the small system of lenses primarily the magnifying mechanism is there. So primary magnification is done by the objective lens, and whatever the image is formed, now it will be further magnified by this eyepiece. So together you can say total magnification of the microscope is done by both objective as well as the eyepiece.
Exactly.
Or you can say ocular lens. So this is the combination of lens which are placed in the upper portion of the body tube, right? This can be 5x or it can be 10x. In the case of objective lenses, you are having too many options. Very first, that is the 4x or 5x. It is the scanning power, you can say, scanning power. Next is the 10x, right? So 10x is said to be the low power. Next is 40x. So it is said to be the high power. And if this 100x, or you can say oil immersion, it is said to be the highest power, right? Oil immersion. So it is used to study the microorganism which are having too less size.
Okay, now we will move toward the coarse adjustment.
So this is the coarse adjustment, or you can say this is the screw mechanism by which the body tubes, or you can say if in the case of this microscope, the stage moves horizontally or vertically, right? And you can say this is the coarse adjustment. So you can say it brings about the wide range of you can say movement, right? So it can be raised or lowered quickly over the wide range to bring the objective in the coarse adjustment, right? So you can say approximately into the focus, right? So roughly you can see your objects. Now this is the fine adjustment, which is different from the coarse. Coarse is from the whole screw mechanism you have to remove, and this is the fine adjustment. So it is the screw mechanism which moves the body tube upward and lower in the limited range, right? So object is brought into the sharp or exact focus. So this was about structure of microscope,
Right. So this is all about function and structure of the compound microscope.